Thank you so much, sir. Moving ahead, the chair of this session, Professor Sharad Kumar Mishra, is a renowned academician, former head Department of Biotechnology, Deen Dayal Upadhyay, Gorakhpur University. Sir is a PhD in studies of protease inhibitors from legumes from Department of Biotechnology, Deen Dayal Upadhyay, Gorakhpur University in 2006. An MPhil from Jawaharlal Nehru University in 2000. Sir is also a life member of Biotech Research Society of India, Trivandrum. Professor Mishra sir is a life member of Indian National Science Congress, Kolkata. With a total research and academics experience of 22 years, Professor Mishra sir has 65 research publications, 10 book chapters. Sir is also a member of editorial board of publications like Archives of Phytopathology and Plant Protection, Taylor and Francis. Vidyavarta, an international multilingual research journal and online journal of proteomics. As a reviewer, sir, you are also working with process biochemistry, archives of phytopathology and plant protection, African Journal of Biotechnology, technology in cancer research and treatment, proceedings of the National Academy of Sciences. Currently, sir, you are working on several projects such as identifying the novel source for the cost-effective production of industrially and clinically imported enzymes, developing a lab with animal cell and tissue culture facility for screening phytochemicals for anti-cancer activities. The list is huge. So without taking much time, I would request Professor Dr. Sunil Singh, Sir, Dean, Faculty of Allied Health Sciences, Mahayogi Gorakhnath University, to kindly felicitate Professor Sharad Kumar Mishra, sir. Thank you so much, sir. Moving ahead, the co-chair of the session, Dr. Sujoy Kumar Dhara. Sir is a well-known scientist in the field of veterinary biotechnology. And sir, you are currently positioned as principal scientist, Stem Cell Laboratory, Division of Veterinary Biotechnology, IVR, Izzat Nagar, Bareilly, Uttar Pradesh. Sir is a PhD from IVRI for your work under Dr. M. Solar at Hebrew University, Jerusalem, Israel, on the topic of molecular biodiversity of chicken population in the year 2000. So you have completed your postdoctoral fellowship under Dr. Nissim Benvensti, Department of Genetics, Hebrew University, Jerusalem, Israel, on human embryonic stem cell biology from 2001 to 2005, which time you have worked as postdoctoral associate and research associate in Georgia and Atlanta, USA, respectively. From 2010 to 2017, you have held the position of Senior Scientist, Division of Veterinary Biotechnology, IVRI, in the area of RNA interference in pig stem cells. And since 2017, you are placed as Principal Scientist at Division of Veterinary Biotechnology, IVRI, in the area of transgenesis with stem cells. Sir has been selected on competition to present in 2 international conferences and has been invited in 25 talks as a presenter. Sir has also published 5 book chapters and 24 research and review publications in peer-reviewed journals. With 2 patents in your name and numerous awards, fellowships, and recognitions, the list of achievements is unending. So, without taking much time of the guests and delegates, I would request Dr. Akhilesh Kumar Dubey, Assistant Professor, Faculty of Allied Health Sciences, Mahayogi Gorakhnath University, to kindly felicitate Dr. Sujoy Kumar Dhar. आगे बढ़ते हुए आज के मुख्य वक्ता का परिचय देते हुए मुझे बहुत खुशी हो रही है। हमारे प्रिय पद्मश्री प्रोफेसर डॉक्टर रामकृष्ण विजयाचार्य बसु भारत के महानतम बायोफिजिकल वैज्ञानिकों में से एक, परमाणु चुंबकीय अनुनाद और आणविक बायोफिजिक्स के क्षेत्रों में अपनी विशेषज्ञता के लिए विश्व स्तर पर जाने जाते हैं। प्रोफेसर होसुर टाटा इंस्टीट्यूट ऑफ फंडामेंटल रिसर्च से सेवानिवृत्त है, जहां उन्होंने एक वरिष्ठ प्रोफेसर के रूप में काम किया और मुंबई विश्वविद्यालय में UOMTAE सेंटर फॉर एक्सीलेंस इन बेसिक साइंसेज CEBS के संस्थापक निदेशक भी थे। सर वर्तमान में CEBS मुंबई में राजा रमन्ना फेलो के रूप में और IIT बॉम्बे के बायो साइंसेस और बायो इंजीनियरिंग विभाग में एक प्रतिष्ठित विजिटिंग प्रोफेसर के रूप में काम कर रहे हैं। IIT बॉम्बे के पूर्व छात्र, जहां उन्होंने MSc की पढ़ाई की, रसायन शास्त्र में, और TIFR, जहां उन्होंने रसायन विज्ञान में PhD की। प्रोफेसर होसुर सर ने रसायन विज्ञान में नोबेल पुरस्कार विजेता प्रोफेसर कर्ट उथरिच और प्रोफेसर रिचर्ड आर अर्नस्ट के सहयोग में ITH जूरिक में पोस्ट डॉक्टर के रूप में काम किया। कई प्रतिष्ठित पुरस्कारों के विजेता, जिनमें पद्मश्री, भारत के सर्वश्रेष्ठ नागरिक पुरस्कार, TAA उत्कृष्टता पुरस्कार, IIT बॉम्बे से प्रतिष्ठित पूर्व छात्र पुरस्कार, CSIR से प्रोफेसर GN रामचंद्रन स्वर्ण पदक और JC बोस राष्ट्रीय फेलोशिप पुरस्कार शामिल है। सर भारतीय विज्ञान अकादमी, भारतीय राष्ट्रीय विज्ञान अकादमी, राष्ट्रीय विज्ञान अकादमी TWAS और ISMAR के फेलो हैं। उन्होंने 251 से अधिक लेख लिखे हैं, 3 पुस्तकें लिखी, संपादित की, जिन्हें दुनिया भर में अत्यधिक उद्धृत और मान्यता मिली है। वह रसायन विज्ञान में चुंबकीय अनुनाद, रसायन विज्ञान के अंतरराष्ट्रीय जर्नल और वैज्ञानिक विश्व जर्नल सहित प्रतिष्ठित पत्रिकाओं में संपादकीय बोर्ड में कार्य करते हैं। दरअसल उन्होंने NMR और बायोकेमिस्ट्री और बायोफिजिक्स से संबंधित कई राष्ट्रीय सम्मेलनों में पूर्ण व्याख्यान, आमंत्रित व्याख्यान, उद्घाटन व्याख्यान और अध्यक्षता सत्र दिए हैं। तो आगे बढ़ते हुए मैं पद्मश्री प्रोफेसर RB होसूर जी से निवेदन करूंगा कि वो कृपया मंच पर आएं एवं हर्बलोमिक्स एंड एमर्जिंग फ्रंटियर विषय पर प्रकाश डालें।
सर हेलो हेलो बोल दीजिए।
हेलो हेलो सुनाई दे रहा है? हेलो
हेलो 1 2 3 टेस्ट। हाँ सर बोलिए।
हेलो
कार्डलेस माइक वहाँ सर वहाँ भी माइक है सर, इसको लग रहा है वहाँ पे माइक भी
आ रहा है? हेलो हाँ हाँ एक पॉइंट और एक पॉइंट First of all, let me thank the organizers of this conference, the Mahayogi Gorakhnath University, and the chairpersons here, all the students here, and it has been a great pleasure for me to come here to talk on this wonderful subject which relates to translational research, especially in the context of Ayurveda, pharmaceutical sciences, which has been the hallmark of Indian science for several thousands of years and had been ignored. And now efforts are on to revive it and take the best benefits of that, not only for India, but for the world at large. It is an opportunity for us to lead the world, be on the frontiers, choose topics which are relevant to human health and global peace. So in that context, this conference assumes great importance. And a lot of young minds here who have come to listen to the various talks surely will get inspired to pursue research in that kind of areas, which will keep our country at the forefront. So in this context, I have chosen to talk here on a topic which is very close to Ayurveda and also close to the modern science. So I said it as herbalomics, an emerging frontier. I will try and qualify both these statements. What is herbalomics and why is it an emerging frontier? So here I have been working at various places, therefore I have all the addresses here. So herbalomics deals with the following topic. Briefly, you see here there are two. On the left hand side, you have the plant world, the animal world. Of course, human beings belong here. And the plant world is all what we deal with, the herbal products, plants, Ayurvedic medicines, all of those. You can hear me, right, with this? If I come out here, can you hear me at the back? Maybe I should stand here only. Okay. Yes, this is. So, so there are two things here. There are two things here. You have the plant world, all the herbs and things like that, we come from here. And our ancestors, rishis and etc., all of them have used these herbs to treat all kinds of medicines, all kinds of diseases. And the modern world is here. And in general, this is a kind of a science which is being pursued all over the world. If you go from here to here, this is what is called genome to metabolome. Modern science people know all of these things. What is the genome? What is the transcriptome? What is the proteome? And what is the metabolome? This applies to both the plant world as well as the animal world. So you go in this vertical direction to study all of these things. The horizontal thing is a connection between the plant world and the animal world. So, and that I call it as herbalomics. Okay. Now, this put it in a little bit more detail. Genome to metabolome goes in this manner. The genome is something which relates to our DNA, what is the size of the DNA and how many genes are there typically. So if you have in the DNA about 30,000 genes is what is one what estimates for a particular cell, particular DNA, and this gets converted into the mRNA. This is the transcriptome, the process of expression here. So this produces 30,000 mRNA, and from this they get translated into proteins. They are typically more than 100,000, including post-translational modifications. And we believe that it is the proteins which are responsible for the biological function. The information comes from here, the hereditary relationships, etc., are related to this. And then the proteome interacts with the metabolome. These are the various metabolites. This comes from various sources. Inside they are synthesized, or you eat food, the food is digested, you get various kinds of metabolites. And they are all being used, and there is a close interaction between all of these stages. And every phenotype means everybody all individuals, they all are characterized by these combinations of all of these things. They are different in different people. Some are common, of course, that is what the modern medicine uses to treat everybody in the same manner. We will come to that a little bit later. Okay, now why is it an emerging frontier? So Ayurveda has been there for thousands of years. People have been doing, any city you go, you find lots of Vaidyas. They give you powders, various kinds of churnas, and you take them, and people are quite happy with it. And this has been working very well. Now, why is it an emerging frontier when we say this? So one point is the following. We had the wisdom in the ancient world, and the modern world is pursuing in a particular way. Now, to make that wisdom available to the modern world, you need to do something. You need to make it acceptable in a general public, to the modern community, scientific community. You have to make it acceptable. So you have to talk in that language. You have to characterize what the ancient people had been doing using the modern tools. So that you have two systems here. One is the modern science system. Other one is the traditional knowledge system, which relates to the Ayurveda and all things which were there in the various Vedas. So using modern technology to achieve new breakthroughs would be globally beneficial. Because you have to talk in a language which is understandable to those who have not read these ancient philosophies. Okay, so you have to talk in that language. You have to use the modern techniques to characterize things in a way that is generally acceptable. And modern techniques are available today. So you make use of these to develop a knowledge which is generally understandable to the common public. So that is what is the challenge. So therefore we say you call it as an emerging frontier, and this science will be there forever. It will take India to the very forefront. So therefore integration of modern and traditional systems of medicine is what is the frontier. Okay. Now to put some ancient wisdom in perspective, it related to all kinds of matters, hence science, health, and profession. Among the various Vedas, as you know, there are 4 Vedas in our tradition. There are Rigveda, Yajurveda, Samveda, and Atharvaveda. Atharvaveda is the one which actually deals with all kinds of matters related to society and human beings and things like that. There are various parts in the Atharvaveda. Out of that, Ayurveda is one of those which is called as Upaveda of Ayurveda. There are various kinds of suktas in a language, in the Sanskrit language. These mantras or suktas which are written in the Upanishads and the Vedas, they are called suktas. This one deals with diseases in medicine. Then you have prayers for long life in this Ayusha Sukta, Paustika Sukta, building houses and agriculture, Prayashchit Sukta, repentance and remedies for wrongful acts, Sri Karma Sukta, love and marriage related issues, Rajakarma Sukta, kings and their duties and responsibilities. So you see, this Atharva Veda actually covered a whole lot of subjects. So there was a lot of wisdom in all of those things. So of course, for the current conference subject, we focus on this particular topic, which is the treatments of diseases and medicines. Okay, there are 3 particular texts which are there, which have become the most kind of source books for all knowledge in this area. These are called Charaka Samhita, Sushruta Samhita, and Ashtanga Hridayam, Pravanavata. These are the 3 fundamental books which are there, on which you learn about everything about the diseases and the treatments which were being given at that time. And now we have to understand that in the modern language. That is the emphasis. Okay, now Ayurveda focuses, there are many experts here in Ayurveda, so I don't want to take too much about that. Of course, they will know this much better than I do. Ayurveda focuses on constitution and inner balance of the body, which is called as the Prakriti. And this is determined at conception. Constitution is determined at conception and remains the same throughout one's life. And this is stated in the same thing. So, and then, of course, how is the body made? Stated here, Sarvam Dravyam Panchabhautikam. There are five elements which are responsible for the construction of the whole body and in the universe, and that is space, air, fire, water, and earth. And there are three important entities which are called as the Vata, Pitta, and Kapha. These are combinations of these five entities which are called as the Panchamahabhuta. And combinations of these lead to various matter present in all creation. Now, every individual is characterized by combinations of these, and this is determined right at the beginning of the conception. Therefore, every individual is different. So, to put it in another language, one dress doesn't fit all. Everyone has his own dress. So therefore, that leads to what is called as a modern people have accepted called as a personalized medicine. We'll come to that a little later. This was already stated in Ayurveda thousands of years ago. So every individual is different. So his treatment has to be individual specific, not that one dress fits all. So although many common things may be there, but nevertheless, every individual is different, and the effects of those every particular medicine is going to be different in different places. And the other important factor is the body, mind, and the consciousness. See, in the modern science, consciousness has no role; it's only body. Now, and the mind, of course, is part of the body. Here you have to combine body, mind, and consciousness. And this is a very important concept which is there in our ancestral wisdom, which one has to make use of to develop it. I will show you where all these things information comes from. Okay. Now, in Ayurveda, as I mentioned, this is a holistic description. Disease is a holistic description. Any misbalance, imbalance in this Vata, Pitta, and Kapha, these three entities which are there, that leads to diseases. So Ayurveda deals it in a holistic manner, treats the whole body, not symptomatic based treatment. Modern medicine treats on a symptomatic basis. You have a particular pain here, okay, treat the pain. Particular pain here, treat the pain there. Whereas Ayurveda treats in the whole body system, it generates the immune system. I mean, treats the immune system of the whole body, of the individual. So therefore, here disease in Ayurveda is viewed as a lack of proper cellular function due to an imbalance of Vata, Pitta, and Kapha, and also of mental condition. Okay, what are the effects? If you look at effects of increase in Pitta, so this is the Sanskrit statement there. Okay, now the meaning of that is here. What are the indications of this? What if they increase pitta? You have yellow discoloration of the fecus, urine, eyes, and skin, excess of hunger and thirst, feeling of burning sensation, and very little sleep. They're all there here. Alpanitrata, Hridaha, okay, Pita Vishnu, Mutra Netratva, all of those things are there indicated in the Sanskrit there. So therefore, it's very important now we have to learn Sanskrit very well. This has been a forgotten language. We need to learn that. Okay, so now effects of Vata, again, see this is a Sanskrit phrase which has been there in the Ayurveda. And what are these indicators? So you see here, emancipation, black discoloration, desire for hot things, tremors, bloating, fullness, distension of the abdomen, constipation, loss of strength, loss of sleep, loss of sensory functions, irrelevant speech, delusion, dizziness, giddiness, timidity. Look at the detail what they had given. If you have this particular fellow which is increased, all of these things will happen. So there are multiple effects that can happen as a result of one particular dosha which is being changed. So there is the imbalance. Similarly, you have for the kapha as well. Kapha it says release of weak digestive activity, excess salivation, lassitude, laziness, feeling of heaviness, white discoloration, coldness, looseness of the body parts, dysphonia, asthma, cough, cold, excess of sleep. And all of these happen because of increased kapha in the content. And that is stated again in this Sanskrit shloka. Okay, now Ayurveda places great importance on the diet because you see now it has to treat all of these things. The diet is most important there. Okay, now because these ones have the effects of balancing these three quantities. So if you have, these are called as the rasa. There are 6 kinds of rasas here. The sweet actually has the following effects. It decreases the vata, decreases the pitta, and increases the kapha. So similarly, amla, the sour, decreases this vata, increases the pitta, increases the kapha. So like that, each one of these rasa, the rasa is an important element in Ayurveda. And this is the diagnostic, and different combinations of these, therefore, help in balancing the combinations of these vata, pitta, and kapha. That's why diet becomes a very important element in Ayurvedic treatment. So apart from the medicine which you take particular kind of a medicine, you also have to maintain a proper diet. So therefore, it has a very synergistic and holistic effects on your body. Okay. Now, disease is, first of all, treating the disease is one thing, but you need to prevent it. Prevention is even better. So the prevention has to be done. Prevention is again dependent on is achieved by diet and lifestyle. This is actually a shloka from the Bhagavad Gita. Yukta hara viharasya yukta chishtasya karmasu yukta swapnamohodasya yogo bhavati dukkhaha. So if you are able to manage your food very well, the diet very well, exercise very well, and you can control your sleep and do the proper karma. This includes the yoga here. For example, yoga bhavati dukkhaha. So therefore, all these elements are already stated in Bhagavad Gita. And Bhagavad Gita, if you know, it is actually supposed to be is the saransh or the summary of all the Upanishads. It is stated in Bhagavad Gita is a kind of a is a science of self-management. How do you achieve your own peace? How do you achieve good health? How to maintain your day-to-day life? So all those things are described in the Bhagavad Gita. So therefore, it is indicated here, what are the components of this particular shloka here? So yoga is a healer, can balance the doshas prakriti. And there are prakriti-specific asanas, and these are the yogas. So, okay, now yoga is not just asana. I think one should clarify here. Don't think yoga means just doing asanas, physical exercises. It is not that. It's called chitta vritti nirodha. Yoga means chitta vritti nirodha. Or samatvam yoga uchyate. That is also the statement. Bhagavan Krishna says in Bhagavad Gita, yoga definition is samatvam yoga uchyate. Keeping a balanced mind. Balance. And Chitta Vritti Nirodha is the Patanjali Sutra. That also says the same thing, that you have to control your Chitta and Vritti and the actions. And the same thing is stated in this Bhagavad Gita shloka. By doing this, it can mitigate all your sorrows, and meditation is an important part in all of these things. Meditation actually consumes a lot of energy. Meditation is not just sitting, close your eyes and things like that. No. Meditation also has a certain way of doing it. You must be able to concentrate very well, not thinking about anything else. If you close your eyes and start thinking about the stock brokers, what is the stock market and how these things, that is not meditation. So you have to have a proper control on your mind, and that is meditation. And this is a common thing for all prakritis. Regardless of what prakriti it is, meditation is required for all of them. Okay, now let's look at it in the modern science perspective. Modern science perspective is talk in a different language. The mechanistic thoughts. What we do here, we talk about the metabolites, metabolic disorders. There are various metabolic pathways in your body, and the food is eaten, it gets broken down into small things, and it goes into metabolic pathways. Metabolic pathways are the ones which actually generate various energies, digest your food, and things like that. And thousands of metabolites which are there, unknown proportions in a complex mixture, unknown metabolites, hundreds of metabolic pathways, many unknown, regulated expression of metabolites, synergy among them, variations due to diseases, variations from species to species, variations within species due to genetic, environmental, and diet variations. This is the language of the modern science. Okay? Unlike what we said, the Vata, Pitta, Dosha, and all of those things, this is the language which is used in the modern science. Now you have to integrate these two. You have to combine these two to bring out a proper knowledge which can integrate the two systems of medicine. Okay. Now, what do the herbal drugs do? Look at the herbal drugs in the modern science perspective. Okay. Herbal preparations cure simultaneously seemingly unrelated metabolic disorders, different diseases. These are called the holistic effects. I will show you some examples of this as we go along. Understand these holistic effects at molecular level. Study interactions of herbal preparations with human proteome. So this leads to what is called a systems biology in the modern language, and all of that leads to mechanistic insights. Then identify possible targets and study specific interactions and synergies to eventually develop better therapeutic protocols. So this is the protocol for the integration outcome. Now let's look at some of the holistic effects. I will take some examples to show you what I mean by holistic effects and what is mechanistic thought. Take for example Triphala. Triphala is one of the most common herbal products. This is actually a mixture of three fruits here, Amalaki, Vivitiki and Haritaki. And this facilitates healthy secretion of pitta in the gut. This balances healthy coating to kapha in the gut. Haritaki ensures healthy movement of vata in the gut. They support natural body functions such as liver, respiratory system, immune system. See, these contain various components. There are many, many compounds in each one of these, and they are able to influence all of these, liver, the respiratory system, and the immune system. Therefore, and health benefits of Triphala, proper digestion, healthy absorption and assimilation of nutrients, which is essential for maintaining a healthy body. Cleansing and detoxification of your body, which leave you rejuvenated. Cleansing of the gastrointestinal tract. Now you see here, this has been realized and the Western world is jumping onto it. You better do it before they do it in a much bigger way. So this is now Triple American Herbal Pharmacopoeia. They have brought out this book. This is a book, a big volume. And you can see here, Triphala is a component of nearly 1,500 Ayurvedic formulations for various different kinds of things. These are mixed with various things, different proportions of the three fruits, and all of them have come out with this many kind of things. And I will show you some examples of this with regard to the effects of those each of the certain diseases. Okay, that is Triphala. Then you have this Ashwagandha. Ashwagandha is another important plant, and its effects are given here. This is a Sanskrit shloka for that one, and what's the meaning is given in Hindi here. Okay, यह वातो एवं कफ शामक सवित्रो शोध और क्षय को दूर करता है. यह बलकारक, अत्यंत शुक्रवर्धक, रसायन युक्त हिता है. इसमें तिक्त एवं कषाय रस होते हैं. So ras is the important word here. The ras I already told you. There are 6 kinds of rasas which are the characteristics of our foods, and those ones are controlled in this manner. And then you have this Vedic medicine. This is called Prashniparani. Prashniparani, this is actually stated in this here, in this shloka in the Atharva Veda. And what does it do? The plant is Pharmacopoeia of India, recommends a decoction of whole plant in alcoholism, insanity, psychosis, cough, bronchitis, dysphonia, diseases due to vitiated blood, gut, bleeding piles, blood dysentery, acute diarrhea. The plant is credited with fracture healing properties. Its total extract exhibits better and quicker healing of fractures in experimental animals due to early accumulation of phosphorus and more deposition of calcium. And this is the plant there. Okay. And therefore, this is an Aushadhiya Suktam, which is from the Rigveda. This comes, and these herbs, so here these herbs and medicines come together as ruling powers and act together. That is synergy. So when I say act together means that is synergy. That specialist scholar is called the physician, who is a destroyer of evil, eliminator, and disease of all. So synergistic and holistic actions of herbs, this is in Ayurveda. Okay. Now another important thing what our Atharva Veda specifies is water is the most important medicine. Jal. See here, this is Apa Idavavo Vaishajya Apa Vivachitini. Okay, you can read that one there. What is it? जल ही औषध है, जल ही सब रोगों का नाश करने वाला है. जल किसी एक रोग की नहीं, समस्त रोगों की औषधि है. हे रोगी, जल तुझे क्षय, कुष्ठ, मृगी आदि क्षेत्रीय रोगों से छुड़ाए. So if you, and all of us say that if you have some problem, you drink sufficient water. Every doctor today also says to drink enough water. But there is also a procedure in drinking water. Ayurveda specifies that. Don't drink cold water, number one. And even when you drink water, you should not stand and gulp water like that from the bottle directly. They say you have this procedure. You have to sit down and drink sip by sip. And then when you are eating food, you should not drink water while eating food. After the food, give some gap and then drink food. So people used to live for hundreds of years, 100, 200 years, people have lived earlier. So, and by simply controlling the water, how you consume your water. So therefore, they say water is the universal medicine, provided it is consumed properly. It is consumed in the appropriate time, in the appropriate manner. Okay. Now, with these things, integrate ancient and modern wisdoms. Modern medicine treats diseases based on symptoms, while Indian traditional knowledge system takes a holistic view where subjects are classified on the basis of prakriti and treatments are personalized. Body, mind, and consciousness work together in maintaining balance. Therefore, it is necessary to develop comprehensive understanding of TKS using modern technology. This is again the word which I'm using because this is the way the frontier is, to achieve a new breakthrough which would be globally beneficial. The steps for integration of modern and traditional medicine: use modern science technologies to establish mechanisms of actions of the Ayurvedic medicines and treatments, enhance credibility and popularity in the modern scientific world, evolve standardization protocols for herbal medicine, keeping in mind the heterogeneities depending on the source and processing of herbal products and supported by clinical trials. This point is important because if you go to the Vaidyas, various Vaidyas give a little bit of the churn, they say, okay, 1 pinch, 2 pinch, 3 pinch, like that. But that is not acceptable in the modern world. You have to give in a proper quantitative manner. How many milligrams? What are the other components present there? What is the dosage? How many times? Before food, after food, when you take it. All these standardizations are required. Quantified, not pinch wise. No, that will not work. So therefore, all that has to be done appropriately here. And update the educational system to integrate the two. Enhance awareness of the benefits of combination of the two practice. Now, how do you in education system you should enhance it? First thing is to learn Sanskrit. So you have to understand what is there in the Ayurveda, because all that is there in Sanskrit. So therefore, one has to learn that one. That has to be quite important target in this sense. Evolve professional guidelines for practitioners of the traditional system. Many times people do not care. Okay, they will say, okay, it is working. Why do you bother? It is working, so I give it to whosoever wants to take it. They will come and things like that. But we want to make it as a generalized system acceptable to the world at large. So therefore, it is not enough if it works in 1 or 2 individuals. That is not enough. Or with 1 or 2 Vaidyas. That is not enough. It has to become a general practice. It is a respectable practice. Everywhere it can be used all over the world. Then you achieve. See, for example, we had this case of turmeric. Turmeric was a drug which was being used for thousands of years in our country, and America gave some medicine. They wanted to take a patent on that one. Fortunately, we objected and won the case. That's fine. But then, see, we have to do this systematically so that it becomes acceptable all over the world. That requires hard work, and that is why I say it is an emerging frontier. Okay, what are the modern scientific tools? Modern scientific tools. So I have carried out research in one of these areas for the last 50 years. So these are the modern scientific tools. Okay, so molecular biology, structural biology. In structural biology, there's X-ray crystallography, NMR spectroscopy, electron microscopy, optical spectroscopy, computational biology, to list some of those. There are others, many more others, but some of those I have listed here. Okay, and the recent paper here, the genomic insights is called as Ayurgenomics here. Genomic insights into Ayurvedic and Western approaches to personalized medicine. So now we see what an integration it is. Ayurvedic treatment, personalized medicine, and genomic analysis. So therefore, it is combining the two. Therefore, it is called as Ayurgenomics. And this paper said, we demonstrate how this is a gene could qualify as a molecular equivalent of Tridosha that can modulate different phenotypic outcomes, where hypoxia is a cause for consequence both during health and disease states. Studies affirm that integration of Trisutra framework through Ayurgenomics can guide the identification of predisposed groups of individuals and enable discovery of actionable therapeutic points in an individualized manner. So this is the point, individualized manner. And you see, there is another paper. There are several, but I've just picked a few. And this says, modern literature, we found that PGM1, this is the kind of a gene, correlates with the phenotype of Pitta as described in the ancient text of Charaka Samhita. So these are not my words, okay? This is just the cut paste. I've just taken this word from there. Suggesting that the phenotypic classification of India's traditional medicine has a genetic basis, and its Prakriti-based practice in vogue for many centuries resonates with personalized medicine. So therefore, this means modern science acknowledgement of personalized medicine in Ayurveda, convergence of TKS and modern science. Okay, so therefore, this they put in the cartoon fashion. Current medicine, one treatment fits all. This is what I said, one dress fits all. And this year, therefore, you have the therapy, some effect, no effect, adverse effects, side effects. But future medicine is personalized diagnostics. So individually, you treat them separately, depending upon their nature, prakriti. Everywhere it is positive effect only. So therefore, this is the way one has to go. Okay, now I come to some of this, our own research here with regard to the objective which I mentioned. Use modern tools to understand the holistic effects of certain herbal drugs. And this is actually driven by one of my colleagues at UMDA CBS, Dr. Chaudhary. TKS investigation using modern tools. These are the fibrils produced by a particular protein called as alpha-synuclein. This is responsible for a large number of neurodegenerative diseases. Okay, Parkinson's disease, Huntington disease. These actually come as a result of self-aggregation or the fibrillation of this particular protein alpha-synuclein. Okay, these are the alpha-synuclein fibrils. Now you treat this alpha-synuclein with 0.5 mg per ml of Triphala. You see, you don't find any of these. What is this picture? This picture is the electron microscopic picture. Electron microscope shows you the details of the topology of the individual systems, and you get the electron microscopic picture like this, where the fibrils are clearly seen. Now you put Triphala in it, you see, you don't find any of those. So the fibrils are not formed. Therefore, you know Triphala works by preventing fibril formation. Okay. Now, how does it prevent fibril formation? We will also see that later. Okay. Now, this is another example. The same Triphala used to treat cancer cells. So here is the cancer cells, HeLa cells, bank 1. Okay. Now you can see here that you look at this picture. This is quite sufficient to look at all of these. There are more quantitative details here. You can see this is the control. Triphala 25 microgram per ml, 50 microgram per ml. And you can see that it is inhibiting the proliferation of the cancer cells. And this is the standard drug which is being used now. And you see Triphala 100 microgram per ml is able to achieve the same one. So the same Triphala, on one hand, is able to control neurodegenerative diseases. On the other hand, it is actually control cancer proliferation. This is what I said in the beginning, that these herbal products have the possibility of regulating the seemingly uncorrelated metabolic pathways. Clearly, the metabolic pathways for cancer and Huntington neurodegenerative diseases are different. But there are components in each of these. They actually work on those and do in a synergistic manner, so they can simultaneously work on both. In a modern drug which you are given, a particular drug which you give, it goes and treats a particular symptom, all right. But you don't know what else it does. It goes and does something else somewhere else. And therefore, that results in various kinds of side effects. And this was done by this another colleague of mine at UMDA CBS, Dr. Manu Lopez. And this is the effect of ashwagandha. Ashwagandha contains lots of polyphenols. And of course, that also Triphala also contains polyphenols, I will show you. And using functionalized gold nanoparticles. Ashwagandha, you make gold nanoparticles, and then actually, this was the ashwagandha is actually put on top of those ones. Therefore, these are called functionalized gold nanoparticles. Okay, now these ones are also able to reduce this proliferation of the cancer cells. And this is the standard drug, which is the Taxol. And then you see the gold nanoparticles functionalized by ashwagandha, and use the different concentrations, you can certainly control the proliferation of the cancer cells. So this is an important development there. Facilitates apoptosis of the breast cancer cells. Now this is garlic, and garlic is an important food, and there are many components in garlic. Okay. Now once again, this also is able to control cancer here. That is why you said in the Ayurveda that diet plays an important role. You need to have a mix of the various components of the food, and if you are taking all of those, automatically natural prevention comes. So therefore, all the side effects will not be there, and you already protect yourself from emergence of the diseases. Okay, now this is another compound which is called saffronol, which is present in saffron. Saffron is, all of us use saffron in various kinds of food preparations, or even during the various pujas and things like that. You use that. Saffron, you see how it controls. These are the fibrils formed by alpha-synuclein. in the presence, in the absence of this safranol, and in the presence of safranol, these ones are gone. They are not formed. Not only that, safranol is able to dissolve the preformed fibrils as well. If they're already formed, then applying safranol actually dissolves it. This again is driven by Dr. Chaudhary, so who is a colleague at UMDA CBS. Okay, now this is a colleague at IIT Bombay. Her name is Dr. Mansi Upadhyay. She actually graduated from, she's a PhD from IIT BHU, and now she is a postdoc at IIT Bombay, and she works on diabetes. And the diabetes here, actually, we have Shilpi Sharma, who is an expert in diabetes, and she works on mycelium encapsulated cytosine nanoformulation for management of type 2 diabetes. And this is the way it goes. They form this, and these are not gold nanoparticles. These are cytosine nanoparticles. You make these nanoparticles, it goes into the intestine here, and it can get released when you change the pH. At low pH is actually compact here, as in the high pH, this is swells, and inside the intestine, this actually gets released when the pH is changed. In the stomach, it is low pH. When you go to the intestine, the pH is normal. It gets released as it goes into the bloodstream. And this increases the bioavailability of the drug, because the drug is inside here now in the nanoparticle. And it increases the bioavailability of the drug and the solubility of the drug, and that's why this nanoparticle is become an important element. Mycelium alone is, of course, also known, but then it is not soluble. Therefore, the bioavailability will be less. But with nanoparticles, it is much better. Okay, now you see she carried out various kinds of in vivo tests also on the rats, and this shows how the blood sugar control is done. Here, this is the initial phase of the treatment, monitoring only for 3 hours, and here it is for the several weeks, several days, four weeks control, and you can see the glucose level is coming down in each of these. This is the without the diabetic vehicle here, and when you treat these with this one's what different amounts, then this will come down. Free mycelium nanoparticle 50 mg per kg, and this is metformin. One of the standard drug which is currently used for managing diabetes, this is metformin. And you see here, it is comparable to the metformin performance. And it is actually controlling the body weight even better than what metformin is doing. Metformin actually increases the weight here, and this fellow is reducing the weight, is producing a good weight on the body of the rats. So therefore, diabetes 2 management, mycelium, chitosan, GLPs control blood glucose and body weight. Okay, so as a result of all of this, we actually wrote a review article in Biophysical Chemistry targeting disorders in unstructured and structured proteins in various diseases. Now we go into the more further mechanistic insights. There we go to the structural insights. The structures of the proteins are like this, various kinds of proteins. They have certain proteins that are listed here. Disorder, misfolding, and aggregation. These are the three elements which are there. Okay, and misfolding can lead to diseases, aggregation lead to diseases, disorder can lead to diseases, occurrence of protein disorder related diseases, then you have misfolded diseases. Now, how do we control these ones? You have to use drugs which actually prevent this misfolding or prevent the disorder or the aggregation. Alpha-synuclein, I already indicated to you one, which is a resultant, it comes because of the aggregation, the fibril formation. And there is another class of proteins which are called as IDPs, or intrinsically disordered proteins, and these ones play quite an important role in biological functions. They themselves do not have any structure in the beginning, but they acquire a structure when they interact with the certain kinds of interactors or some drugs or whatever. So therefore, these are very important proteins, and if you can target these ones, you can control many diseases. And the herbal products can be targeted over all of these. The small molecules, drugs, components can prevent misfolding of protein by stabilizing the native fold. In order to be able to work on this, you need to know the structures of the proteins. And this is an example here, the prion diseases, which how this misfolding of the protein can lead to a disease. This is a functional protein, there is no problem with this, but this gets misfolded, many of these helices are converted into beta sheets here, and this is non-functional, and this actually becomes a disease-prone protein. And this discovery actually led to a Nobel Prize to Stanley Brusner in 1997. Okay, now neurodegenerative diseases, electron microscopic images of the A beta immunoglobulin light chain variable domain, and CACH3. Okay, now this is the result from solid state NMR. From solid state NMR, nuclear magnetic resonance spectroscopy, you actually obtain the structures of the fibrils to a great detail. So how the fibrils are formed, what is the individual structures there. Now if you know the structure, then you know where to target, how to prevent it. Okay, so this is the modern approach in understanding the fibril formation and its prevention. Okay, and here comes the various development of technologies to determine the structures. Therefore, you need to determine the structures. A lot of work, decades of work has gone in there. And one of our students here, this is Dinesh Kumar, who is the president of this society, and he's yeah. So this was the review which was written. This thing was somewhere here in 2012. As a result of various developments here, the technique developments, NMR methods development, the software developments, you can actually now obtain the backbone fold of the protein in 2 days. I will show you that there are further developments which have happened. They're very much in the next slide. You can obtain the fold in 2 hours. This is very important because you need to know the structure. There are 100,000 proteins in your system. If you want to take 2 years to determine the structures of one protein, then of course your lifetime is not sufficient to do anything of that sort. So you need to do things faster. The structures have to be determined faster. Therefore, you need to develop methods which will allow you to do that. And then you study the interactions of those, your drug molecules with those at specific points, which will allow you to establish a mechanism for the action of the drug. Now here is the calculation. This is the human ubiquitin structure, chicken SS3 domain structures. These are calculated from NMR chemical shifts here. And you can see the structures are very well determined to very high level of precision compared to what was previously done by X-ray crystallography. Now here is a development which happened sometime 2 years ago, 2008, not 2 years ago, 3-4 years ago. And this was a method which was developed by one of my postdocs here, Veer Mohan. And this is a very good NMR spectroscopist. Here you can see 3D experiments here, NMR experiments, and in 2 hours, you get this, all the data which is required for structure determination in 2 hours. Three-dimensional NMR experiments obtained in 2 hours. And the calculated structure is shown here. Therefore, that opens up great possibilities for structure determination, understanding misfolding of the proteins, and therefore targeting this misfolding pathways. Okay, now what does AAA do with regard to the cancer cells? Dr. Manu Lopez, he actually carried out various spectroscopic investigations there and determined that AAA alters the secondary structure of tubulin. Tubulin is a protein inside the cell and which is responsible for cell proliferation. And this is AAA alters the secondary structures of tubulin and prevents, therefore, the proliferation of the cancer cells. We will need not go into the specific details here. The title is already giving you the mechanism. Okay, and this slide is not very clear because the light is actually blocking there. So these are the GNPs, AAA coated gold nanoparticles. And using the different controls here, the control and GNPs 350 microgram per ml, 700 microgram per ml, and it shows how the cancer cells apoptosis happens. Their proliferation is prevented, and this is a standard Taxol. And these ones actually, this is at 50 nanomolar, and these are microgram. But here you cannot talk about in nanomolar because these contain multiple components. Okay, therefore you have to talk about the microgram per ml, and that is giving you a mechanism as to how it is controlling this cancer cell proliferation. Okay, and for this is the recent one, which is one of the students here, Mandar Bopatikar, and he actually worked out the further details of the interaction between alpha-synuclein, the small components, and alpha-synuclein protein, understand the fibrillation process. So here, this is the NMR spectra. Well, we will not go into the details of the NMR spectra there, but what it shows you is the following. That is, in the absence of the AAA, the protein is going through the aggregation process here. Okay, and these are areas which are coming close and they are interacting with each other to form the aggregation, and this goes into the kind of a fibril. In the presence of AAA, this aggregation is prevented because it interferes with these cylinders which I am having here. Therefore, it remains more like a monomer. The protein remains like a monomer. Therefore, that does not aggregate. Okay, so this is the mechanism. This inference is derived by analyzing this spectra. Therefore, you know at the residue level detail, the protein's residue level detail, what is interacting where and how do you prevent it? This, because the structure is known. Therefore, the drug can go and interact there. Those are the residues. Specifically, if you see, these are all threonines here, and the threonine amino acid residues, the OH group in the side chain, all the polyphenols in your triphala, they also have OH groups in, those molecules. Therefore, they go and interfere with the OH-OH interactions which are responsible for aggregation. Therefore, it prevents aggregation. So therefore, you have the mechanistic insights there. Okay, now this is, then it took three different molecules here, three different individual molecules, which are components of that to understand their relative effects. And once again, all of these are able to prevent sulfur-synuclear fibrillation, and this is shown here. And tubulagic acid is the most successful one compared to these ones. These are also reducing the fibrillation process, but this tubulagic acid is quite powerful in preventing the fibrillation of the protein. And this is the NMR data, which actually shows you which portion of the protein is responsible. See here, these are the areas which are interacting with the protein, the areas of the protein which are interacting with the drug. Okay, now this is with regard to the safranol. You see here, in the absence of safranol, the spectra change like this. This is the original spectrum. As the protein starts aggregating for fibrils, the number of peaks will start getting reduced, and you see very few peaks here. Okay, this is an indication that you got the fibril formation. The disease is on the way. Now, if you put safranol, the structure of the safranol is here, and you see this remains here even after 100 hours, all the peaks are still retained, which means it is preventing the fibrillation of the protein. So these mechanistic insights which are there. Okay, now this, I've covered a lot of material here. A lot of students for several decades have worked on these ones. This is the whole list of all of those ones there. And Krishnamon is also here. Dinesh is here. And those ones, of course, and Mansi is here in the super. Yeah, there's so many others are there. But I have a few pictures there. And these were the various students who were prepared. Dinesh is here. Neil is in Delhi. Varshadosh is here. Krishnamon is here. Jeet. All of these have contributed to the developments of methodologies, structure determinations, and study of those things with the things comes from the various groups. Here, this is the group at CBS. This is Shinjan, whose picture I showed you. Anushri, Vaibhav Shukla, Kavita Rachaneni, Sunita Patel, and Veer Mohan. These are all experts in different areas. One is expert in NMR, other is expert in biochemistry, other expert in computational biology. All of these is put together, you start understanding the effects of herbal products on protein interactions. Okay, now this is the group in IIT Bombay, and this is Mansi here, and this is Ashutosh Kumar, and the students in IIT Bombay who have been working on these ones. So various NMR centers which have been used for this, the TFR NMR center here, the 800 megahertz NMR. There is also NMR center at IIT Bombay, Department of Chemistry, and TEM facility at IIT Bombay. So I think these people here which have been the who are maintained these NMR machines at TFR, and without this, of course, you could not have done any of this high field research. Okay, with that, I stop. I thank you very much for your attention.
Thank you so much, sir,
Yeah,
for sharing your knowledge and experience on ergonomics. You have wonderfully correlated different things with one another. So your word that every individual, every patient is different, so treatment varies accordingly, will surely help these young minds to work in that field. And sir, we will surely work on learning Sanskrit. Now moving ahead.
So if there are any questions.
Please tell your name and ask.
Who is it? Where? Ah. Yeah.
Hello, sir.
Yeah.
Good morning, sir.
Good morning.
Myself, Shweta from Allied Health Science. Sir, as usual, you tell us about AAA is very useful in cancer cell. Sir, you also tell us about prion disease. But how can these prion disease can be cured and symptoms about anything about prion disease you can describe us?
See, we have not done experiments on prion disease. I gave it an example as to how the misfolding of the can you hear me? How the misfolding of the protein leads to the prion disease, to the disease. That's one example I showed. You need to do research. You need to do test with the kind of system with that protein. You put a target herbal product, you put it against that protein. You see whether it prevents the misfolding. We showed you the Triphala is preventing fibrillation of alpha-synuclein, right? Likewise, you should see whether or any of these herbal drugs does anything with the misfolding of the prion protein. That needs to be done.
How will we detect this?
By these methods. I showed you the NMR methods, crystallography methods, and my electron microscopy. These methods one should use. There is a whole protocol there for studying protein folding by NMR. NMR is a very powerful technique for studying all kinds of things. Protein structure determination, protein folding, the kinetics, the dynamics, all kinds of things can be done with the NMR technique. Therefore, that is the modern tool which you should use to understand the mechanistic details as to how the protein is behaving in the first place and how the interactions happen with that protein, with the drugs which you choose, which components of the drugs are playing what roles, all of that you can figure out.
Thank you, sir. Sir, I have the second question.
Yes.
Sir, but it is not related to that topic. As usual, in starting you tell us that every person does not fit to every each other. And as usual, we need specific medicine for each individual. So how can we achieve this thing? As I know that merging both herbal things and the modern technique, we are still not far for that achievement. So
yes, that is true. That is where the research is required. This is why I said this is an emerging frontier. That's why I qualify that statement as an emerging frontier. We need to study the individuals carefully, study individuals properly, analyze their proper behaviors. Many of these things you determine by analyzing their behaviors, their body structures. So the persons who have certain properties of Vata, they have different kinds of body structures. Obesity or various different things which are there, or somebody is always a thin man, no matter how much he eats, he is always very thin. But somebody who doesn't eat anything becomes so fat. So these are all dependent on the structures of the system, of the individual. So you have to study that one very well. Okay, then only you can make personalized medicine properly. The components, see these various components which are there, the triphala, the three fruits of which are mixed, their proportion can vary. It need not be the same proportion always. This can vary from individual to individual. So that only the Vaidya will determine how much one should add of each one of those, and quantitatively and prescribe it and document it so that somebody else also should be able to use it afterwards. So today you take a paracetamol tablet, okay, you don't go and see whether this paracetamol tablet is this much or that much. You just take it, right? Because it is already standardized. So we need to develop that kind of a standardization procedures for these individual specific things, components. So you have, for example, amlopin drug is there, which is used for controlling blood pressure. You have 2.5 mg tablets, you have 5 mg tablets, 10 mg tablets. Why are they made? Because they are required for different kinds of persons. Depends on the level of aggravation of the heart problems. So likewise, one should prepare these various kinds of proportions, tablets with different proportions. Then it can become personalized.
Thank you, sir.
Any other doubts?
Yes. Yeah, there is here.
Good morning, sir. Myself, Ruchi Sukla, BSc Biotech, third semester. Sir, I want that you describe the neurodegenerative disease. Sir, please tell about
Neurodegenerative is all these Alzheimer's, Parkinson's, Huntington's, all of these are neurodegenerative diseases. Basically, what happens is your brain is affected. That's why they are called neurodegeneration. Okay, brain cells are affected, so the brain doesn't function properly. So they lose balance, they don't have control, the memory gets lost. All of these are because of degeneration of the neural system. So therefore, you have to prevent that. The reason for that is, as you understand, this one particular protein is responsible for all of those things. Some people say it is a deposition of the fibrils in the brain, which actually affects the brain components. Some people say it is the oligomers of the oligomers before the fibrillation happens. They are toxic to the neurons. Okay, now you have to see how the oligomer formation can be prevented. Okay, so you have to target those. Once you understand this mechanism, then you target those, the prevention.
Thank you so much, sir, for your responses. If any
Yeah.
students having any questions, last, please stand up.
Excuse me.
Where?
I need to wind up. Maybe very quick.
Yeah. Yeah.
Sir, just 1 minute. I have one little query.
Uh-huh.
Good morning, sir. Thanks for your excellent presentation and excellent work. Sir, just during the most of the in vitro experiment, we faced the problem regarding the solubility of the most of the drugs and pH also. So how to take this problem? How to tackle
That
this
is where these carriers, nanoparticles come into the picture. This myrcene, as I showed you this one particular example in diabetes, this is not soluble. So therefore, its bioavailability becomes less in your bloodstream. Now, if you make it through not gold nanoparticles, cytosine nanoparticles, its solubility increases because it provides a surface which is hydrophilic, and the drug is put inside there. So, because of the hydrophilicity of the surface, its solubility is increased. But now this is also pH sensitive. Therefore, if we increase the pH, the nanoparticle opens up because we need to have delivery also, not just go inside. It is to be released also. That means, therefore, it has to be broken. Therefore, at high pH, it gets broken, then it is delivered. So these are the tricks one has to use.
Okay. So Professor Hulsud is available. Whosoever has got any more questions, interact with him at personal level, not at this point. Okay. Thank you very much.
Thank you so much, sir. Now,
Okay, thank you. Oh.
I would request the chairperson and the co-chairperson to kindly felicitate the esteemed speaker with certificate and award.
Thank you. Thank you. Thank you so much.
It was wonderful.
Thank you so much.
We learned so much.
Thank you so much.
A huge round of applause for sir.
Thank you.
We'll be in touch, sir.
Sure, sure.
Thank you so much.
Thank you. Great meeting you. Yeah. Yeah. Great to meet you.
Sujay Dhara from Indian Veterinary Research Institute, IVRI.
Great.
Moving ahead with the session, Dr. Madhav Neelkant Mogale, Principal Scientist and Associate Professor, Department of Toxicology and Experimental Medicine, DSIR, Central Drug Research Institute, CDRI, Lucknow, is the speaker of the next lecture in this three-day conference on the topic of exploring the hepatoprotective and nephroprotective potential of natural compounds. So you are currently working as a senior scientist since 2017 at CSIR Lucknow and as an assistant professor since 2019 at Academy of Scientific and Industrial Research, Ghaziabad. A PhD in veterinary pathology in 2015 from Madras Veterinary College, TANUVAS, Chennai, Tamil Nadu, and MSc in veterinary pathology in the year 2011 from College of Veterinary Science, GATVASU, Ludhiana, Punjab. Professor Mugale sir has extensively worked in the field of regulatory toxicology, such as GLP in vitro studies, execution of regulatory toxicity studies as per OECD, EPA, ICH, EMEA, ISO, new drugs and clinical trials, toxicopathology, veterinary pathology, experimentation on non-human primates, and nanotoxicology. Respected sir has been awarded with several awards and fellowship. I feel really privileged to call you upon stage for your lecture. Please welcome Dr. Mogale sir with a huge round of applause.
Thank you. Let me thanks the management of Mahayogi Gorakhnath University and organizer committee, Sunil Kumar sir and Dinesh sir for inviting me. My today topic is exploring the hepatoprotective and neuroprotective potential of natural compounds. In my lab, we are specially working on the liver carcinogenesis, hepatotoxicity, So the natural compounds having long history. Last 2 days we are listening the same. There are many plants are usually in the developing and developed countries, especially in developing countries. These are the Ayurveda medicine commonly used, and globally 17 to 18% of plants are currently utilized for the medicinal purposes, and about 150,000 plants are reported to contain some secondary metabolites that are also toxic to the humans and animals also. So, globally, 80% of people use plant derived products, particularly in the developing countries. So, effectiveness and tolerability of the medications are now used to treat and also the serious side effects. And nowadays, a lot of interest is coming in these natural compounds because of the toxicity of the synthetic or single molecules and to reduce the secondary side effects of the allopathic drugs. So, growing interest has resulted in the investigation of natural plants having the potential medicinal properties, including ginkgo biloba, mycorrhiza coa, and cannabis tenuspora, etc. So, I'm going to talk on liver and neuro only. And in my lab, we are working on the liver. Liver, especially, everybody knows the liver is the largest organ of the body, and it's very important organs of the body. It plays a very significant role in the metabolism and excretion. It controls the carbohydrate metabolism and also having the secretory and hormonal balance roles also. So in the nowadays the in the market, if you look the data of the liver problem, there are near about 52% people of the society having the fatty liver, either in the grade 1 or grade 2 or something like that, due to the lifestyle and as Dr. Ozusa told, the lifestyle and diet of the person. So nowadays, sedentary life is more in the, especially in the urban areas. So in India, there are about 52%. You can just imagine how much they are. The liver affection is there. And the marketed one commercial product is also available. If you see in if you go in any clinic here, you will give the better protective, or you can say the better stimulant, appetizer, liver 52, liver 52, marketed by the Himalaya company. So there are many plants such as Artemisia, Woodfordia, erigoid glycosides, isoquercetin, and glyceriza. So every plant having, there are many constituents, but main constituents of the responsible for hepatoprotection, they are also isolated. So if you see ginkgo biloba, actually I had not worked on the ginkgo biloba, but there are many papers says that the ginkgo biloba extract treatments for 8 weeks in carbon tetrachloride induced liver injury and fibrosis reduces the oxidative stress by inhibiting the mechanism NFKB and hepatic stellate cells, an expression of the TGF beta 1. These are the markers used commonly for the liver injury. So if you see in the liver diseases, there are many diseases are there, such as swollen liver, fatty liver, jaundice, liver toxicity, liver fibrosis, liver cancer, viral, bacterial, and drug-induced liver injury. So all these, how they are diagnosed? By the liver function test. Not all the tests are important for the diagnosis of the liver problem, but it indicates the injury to the cells, induced cellular enzyme production, influence on the enzyme half-life, decreased catabolism, or removal. So when we will go to the clinic, the doctor will prescribe some tests such as liver function and kidney function. They are having different thoughts in the mind. As per that, they will advise for you. So the tests are such as AST, ALT, SDH, and GLDH. And more specific test is ALT. ALT is a more specific and sensitive biomarker for hepatocellular injury in rat, dogs, and non-human primates.
increased serum ALT actually need to be specific for hepatocellular injury, and enzyme induction increased hepatic intercellular concentration of the enzyme, store damage and tissue damage, also in the skeletal muscle damage also it indicates. So next test is the AST. AST is a non-specific, but still in correlation with the ALT, they will ask for the test, and it has high activity for the red blood cells, and increases or decreases, it gives the idea for the liver health. So in the pathological data, if you see the correlation of different tests such as ALT, AST, ALP, GGT will give the different organs having the different problems, such as AST will give the liver, cardiac and skeletal muscle abnormality, LP liver and bone, and ALT liver, cardiac muscle, skeletal muscle, GGT liver, cardiac and pancreas and kidney. Sometimes negative effect also there, decrease in AST, ALT, reduced hyperstellar synthesis or release of enzyme, inhibition of enzyme activity. ALP is a marker, especially for the bone strength and also calcium phosphorus metabolism. If there is a decrease or increase in malabsorption of the calcium phosphorus, intestinal ALP decreases with fasting, bone ALP isoenzyme in the end. So GGT is the marker especially for the liver or cardiac abnormality. Serum GGT activity in normal rats and mice generally very low and maybe below the level of detection. And sometimes you can say if you see the GGT 0 to 1 IU per microliter will come. So if there is an increase, there is a problem in the biliary pressure, hepatocellular hypertrophy or cardiac effect. Cholestasis. If there is an increase in the serum bilirubin, that means it's the bile acid got blocked. So increase in serum bile acid, increase in ALP and GGT will give idea. Non-enzymatic parameters such as serum, urine bilirubin and serum bile acid, serum albumin and globulin, plasma coagulation. These all will give the liver health. So the concentration of the bilirubin and the presence of these markers will give the idea of the liver, such as if you increase in the bilirubin, that means there is a yellowishness of the mucous membrane, and also you can say the jaundice of the person. So increase, it might be due to RBC's hemolysis, but sometimes indirect bilirubin we have to see if there is an hemolysis is not there, still increase. So that means it might be due to the not due to the red blood cell lysis, but due to the liver problem, hepatocellular injury, bile retention, altered metabolism compared with the ALT analysis. Serum total bilirubin concentration is relatively insensitive test. So depend on the presence of the bilirubin, direct or indirect, we can say that either there is a hemolysis or hepatocellular injury. As I told you, the cholestasis or bile acid increases due to the bile acid is important, very important for the cholesterol metabolism. If there is a cholestasis, there is an increase in these increase in the serum bilirubin and bile acid also, and decreases the hepatic increases the cholesterol and decreases the bile acid synthesis. Albumin to protein ratio is very important. Liver is responsible for the protein metabolism, and also so 80% loss of functional hepatic mass before hypoalbuminemia apparent, seen in chronic liver diseases, and alter the prothrombin time, prothrombin activated time, and clotting factors also. So liver is very important. So one of the natural compounds work also we had done in our lab. So if you see, this is healthy liver, Steatosis is accumulation of fats in the tissue, liver tissue, liver parenchyma, and if there is an inflammation due to some toxic elements or anything which is damages to the liver hepatocytes, and if there is a chronic, these damage leads to the fibrosis, or you can say after carcinogenesis also. So these steatosis, inflammation, and hepatocyte ballooning will lead to the irreversible liver damage, that is the fibrosis, or in human is called as cirrhosis, and also leads to the cancer. So we had worked on these some molecules. We explored the Vidania coagulans having properties of antifibrotic activity. What we had done, we had took the fruits of the Vidania, paneer, it's known as paneer full or Rishagandha, and it has having many properties. One of them we tried to show hepatoprotective, anti-inflammatory, anti-hyperglycemic, antimicrobial. All these properties are there, but we are only focused on the liver protection. So it contains, it is a phytosteroid and modified steroid lactose such as duolinolidis. It's a major compound. This Vidania coagulans fruits, we are extracted in it hydroalcoholic extract, and from there different types of steroids we are isolated. So this molecule K1013 showed better activity in the liver fibrosis. What we have done, we took the Rex2 cell line of the human stellate cells. Human stellate cells are the stellate cells. These cells, round oval cells, presents around the you can say bile acid around the central veins. We had isolated the cells, and also we had done some experimentation, and we, with the help of in vitro activity treatment for 48 hours, shown the a good antifibrotic activity. And also we have tried in the HEP-G2 cell lines the anti-steatotic activity, lipid accumulation activity in the HEP-G2 cell lines. In vivo, what we have done, there are two models we have developed by high methionine and choline deficient diet. We are given the high fat methionine and choline deficient diet for 8 weeks, and the diet induced the animal's weight, also induced the fibrosis. So by histopathologically, just I want to show how we had evaluated the liver abnormal. This is the histopathology, HID study. This is standard drug. This is our tested drug C002 and K103, and this is the marketed Picrolio also. So in the liver, if you see the chronic inflammatory cells which we had observed, but there is a decrease in the chronic inflammatory cells in the Picrolio and also in the our K103 molecules. With the help of picroseries staining, spatial staining, we can tell that if there is decrease in the fibrosis, the number of these redness give the fibroblastic cells around the central veins and in the hepatic portal. If you see in this reduction, and there are two markers we are seen with the help of immunohistochemistry, such as alpha SMA collagenase 1 alpha. So with in the K103, there is a decrease in the alpha SMA, smooth muscle actin, and collagenase 1. So other also parameter we had in number of inflammatory cells, and also relative alpha SMA expression, and also relative call1 by real-time PCR. So there is decrease in the these expression of the fibrosis. This is the real-time PCR expression of the different biomarkers, TNF alpha, Pol1, TGF beta, and tissue injury molecule and interferon gamma. So these are the decrease in the treated groups. We have calculated the NAS score, and if you see, our treated group, the ALT, AST, serum cholesterol, and triglycerides, these are the very specific biomarkers used for the diagnosis of the either liver steatosis, you can say, or fatty liver, you can say, or fibrosis, and these are the decrease in the treated groups. Next is a neuroprotection. Everybody knows the brain is a very important, plays significant role in the hemostasis of the body, and it controls lot of functions of the body, and it gives the memory, and it's a, you can say, there are many drugs which are having neuroprotective, memory enhancing, and anti-aging properties. So,
sorry to interrupt you. You just have 5 minutes more.
Okay. Thank you. Thank you. So there are neuroprotection we have not worked, but there are some drugs are there like Tinospora cardifolia, Vidania somnifera, Stereospermum surveillance, Panax ginseng. So there are many diseases are also brains, neuronal degenerative diseases, meningitis, Alzheimer's, Parkinson's, infectious disease, epilepsy, aging, protein aggregation, inflammation, oxidative, and neurotoxicity. So every disease is having their own mechanism of actions, aggregation, inflammation, and etc. So there are different tests are also there, invasive, non-invasive. So non-invasive means neuronal examination, CT scan, Doppler. Invasive means biopsy and CSF collection. So there are many drugs are also there, and they are having different targets. In the drug discovery, you have to first identify the molecules, isolate it, screen it, optimize it, and develop development of the lead molecules. So there are different cell lines are also there for liver cancer and brain cell lines. And in my lab, there are 2 cell lines are there, LX2 and FG2. So we can work. You can work on these cell lines also if you are interested. For designing the assay, you have to do the in vitro activity such as MTT assay, that is cell viability assay in the particular disease. So when you wish to do the drug discovery, you have to go for either in silico interaction, in vitro, and then in vivo models. And then if you are getting a proof of concept of the lead molecules, then you can go for the toxicity and the higher animal study. So different models are also there. So you can go with the liver cirrhosis model, liver cancer model, genograph, diethyl nitrosamine induced aflatoxin, subcutaneous orthotropic, and patient-derived genograph model in carcinogenesis. So similarly, in the brain also there are different models. Whatever the models you want to wish you area, you can do. These are the models for the brain diseases. What we have to do? Select the as per your choice, neurodegenerative or liver toxic liver models. You can select the models, and according to that, you have to give the animal dosing, cure, and you can protect the brain and liver. So when your drug is good enough to get the proof of concept of the going to the market, you have to do the pharmacokinetic study. That means pharmacokinetic study will give the idea of the absorption, distribution, metabolism, and excretion of the particular potential drug. This is very important. If it's getting detected in the blood, that means it's circulating, and if particular tissue is getting, that is it is distributed in particular tissue. So in the drug discovery, these are the steps very much important, such as selection of the lead molecule, phytoconstituent analysis and biomarker identification, in vitro screening, In vivo screening, PK and PD studies, toxicity studies in two species, rodents and higher animals. In the rodents also you have to do rat and mice, that two acute and repeat dose toxicity. And higher animals, any clinical trials, if it works better in this area, then you can go for the clinical trials, phase 1, 2, 3 and 4. So in the natural medicines is a very challenging area. There is a very complex things are there. So complexity is identification of the major compounds, detection of the biomarkers is very important. And if you are selecting the particular things, availability of the natural compound is also very important, and standardization of the molecule. So this is my last slide. So just keep in mind some points. Selection of compound is very important. Perform the dose response curve. You have to do the study in and 3 doses, not the single dose at 100 milligram per kg. It will give the. So when you want to do the real trial and to bring the market, you have to do it on the 3 doses. Especially, we will do 25, 50, 100. You can select, but don't go beyond the 100 milligram per kg. You can use lesser than that. Otherwise, the dose of this one will go 1 gram, 2 gram. Nobody will take in the market. So adjust the doses based on the body weight. Determine the frequency of treatment. Consider the administration, either oral, intravenous, intraperitoneal. But in natural compounds, there is no intravenous. You have to be route of administration is very important. Monitor the signs of toxicity and needs to determine the margin of safety. This is very important, margin of safety. Otherwise, the DCGI will not give the support. Thank you.
Dr. Mukherjee is available over here, so we will not take up. We have already exceeded our time limit. So he is available. You ask your questions, save your questions, meet him, and ask your questions direct. Because we really don't have any time.
Okay. Thank you. Thank you.
Thank you so much, sir, for this wonderful session. I request the chairperson and co-chairperson to kindly felicitate esteemed speaker with certificate and moment.
Thank you, sir. Thank you. Thank you.
Very nice.
Thank you, sir.
Now we'll break for tea from 11 to 11:30. I request all of you to kindly come again at 11:30. Now before breaking, the left side of the stage